This is lecture exam 1 review. We're going to cover chapter 12, chapter 15, and chapter 17. So in chapter 12, the general and special senses, these may not be on the test per se, it's a matching or anything, but you need to be aware of what a chemoreceptor is, a nociceptor, a mechanoreceptor, photoreceptor in particular, because I may use those in a sentence, but again, it's not going to be like a matching or anything like that. If I'm skipping through the slide, that means it's not on there. Proprioception, those of you that had Biology 137, particularly in person, and even if you had it online, you had proprioception was defined on something called the chart in chapter 10 and 11. Proprioception is the awareness of your body in space. If you were in the classroom, I would say if you're sitting at a desk, your foot is probably under the table, and even though you can't see it, you know where your right big toe is. That is proprioception. The rest of these we'll talk some generalities as we go through. When I say in class, these are all mislabeled, in my opinion. You should put the word mechanoreceptor up there, because that's what these are, and you should X this out. Free nerve endings are not considered mechanoreceptors for what I'm teaching you. There may be some that are, but they're pain fibers. So we're going to just X that out. But you should know what a Meissner's corpuscle and a Pacini corpuscle is. Meissner's are for light touch. They're found up in the layer of the skin, the papillary layer, which is where the dermal papilla is, in the top of the papillary layer. In fact, they're actually found in the dermal papilla, if you remember that from chapter 6 and 137. These are found down in the reticular layer of the dermis. You don't have to know that specific location, but these are for pressure, deep pressure. But these are both mechanoreceptors. And while I'm on that, a mechanoreceptor is termed a type A fiber. I get something that shows up here a little better. Mechanoreceptors are type A. Gosh, these are all not showing up. Let's see. Let's see if we can get a color that will show up better here. We're going to compare and contrast these with a pain fiber. Type A fiber is what a mechanoreceptor is. They are considered to be fast. All nerve impulses are fast, but we're talking milliseconds here, which is important. So these are faster than what pain fibers are. They are myelinated, meaning they have that fat coating that we talked about in 137, the myelin coating, which helps it transmit faster. And they are large diameter. Now I'm going to go ahead and skip the pain fibers, which we're going to see here in a minute. These are mechanoreceptors. I'll just put mechano. Pain fibers, in contrast to these, are called type C, or just C fiber. They are quote unquote slow, or slower than a mechanoreceptor. They are unmyelinated. This is why we marked out the free nerve ending. These are called free nerve endings or naked nerve endings because they lack myelin. And these are smaller diameter. So that could be something you may need to compare and contrast on the test, the difference between a pain fiber and a mechanoreceptor. Okay, let's go on to the next here. Again, just to review this from chapter 6 last semester, in the dermis you have a papillary layer, which is about the top 25%, and you have a reticular layer. Up here is where you see not only your free nerve endings, but your Meissner's corpuscles, and your Pacini corpuscle is found down in the deeper layers, even into the hypodermis. And those are for pressure. Temperature receptors, I'm probably not going to have on the exam. Just be aware that you have those. Some may read warm and cold, and then if you get too cold or too hot, then you have pain initiated. Now, here's why we marked out the free nerve endings on the mechanoreceptors, because we're going to associate those with pain. So this is the nociceptor from the page we defined those on. So you might want to write nociceptor up here, or nocioreceptor. Again, these are pain fibers. They are C fibers. They are slow, considered to be slow, unmyelinated, and smaller diameter. They adapt very little, if all. That's very important because when you have pain, pain is your friend. It's telling you something is wrong. This is why it doesn't adapt. It's trying to tell you something's wrong. Okay, visceral pain, we're going to talk about this in two different ways. In this way, we're going to talk about referred pain, which you see at the bottom. Usually on the test, I will focus on the distribution of the kidney and the liver and gallbladder. So if you have a gallbladder problem, like a gallstone or a hot gallbladder, you will see pain over the area of the liver and gallbladder in the right upper quadrant. You'll see it in the flank on the same side, a small part in the back, and you'll also see it in the right shoulder area. So know those three associated with the gallbladder. The kidney and the liver, I guess. The kidney is the largest referral pattern. So where you see the skin-colored chaps here this guy's wearing, the kidney can literally hurt in the largest distribution of any organ. We would do the heart, but I do not like the way they've got the heart diagram. I think it's wrong, but just know those two primarily for your exam. We'll skip that. We're going to skip this. If you did have me for 137, we gave you the chart that had all the areas, functional areas of the brain. And I didn't make you know them then, and you don't have to know them now, but hopefully you made some of those connections between different areas. But you can see here pain, the thalamus was a sensory relay area that we talked about in 137, but it's going to show you some of the associations like the limbic system, and it's going to end up being interpreted in the cerebral cortex. So those were covered last semester when we didn't make you make those connections. It's a little above our level of study here. Okay, here's proprioception. I want you to mark out the Pacinian corpuscle. We just called that a mechanoreceptor. This should not be here in my opinion. But muscle spindles and Golgi tendon organs are types of proprioceptors. If you had me in person, I would do and even on the online, you could probably make this out. When we did reflex testing, somebody hits your knee with a hammer and your knee kicks. What you're doing, you're actually stimulating the Golgi tendon organ, which can cause a contraction in a muscle. If you stimulate the muscle spindle, it'll cause relaxation. So just kind of know that. These help to tell you if your proprioception is good or not. Okay, visceral senses, we just said something about referred pain. This is talking about your sense of fullness after you eat, something called the satiety center. These will probably not be on the test, but it's another discussion of these senses that are in your body. Okay, I'm going to go through the special senses pretty quickly. This had more information on it than the nervous system when I first started teaching this, so I think I narrowed it way down on your PowerPoints, but just kind of look at what I'm specifically giving you, because I'm trying to narrow this down for your testing purposes. Okay, the sense of smell you should know is olfaction. That term, olfaction, is the sense of smell. These are, when we talked about the different types of neurons last semester, we talked about unipolar, bipolar, multipolar. All these special sense neurons are bipolar. That's not going to be on your test, but we kind of set those aside till this semester. You can see these bipolar neurons extending down into the nasal mucosa here. So when you smell, it's a chemical, a chemoreceptor that is reading that, and that will ultimately go into your temporal lobe to be interpreted. Again, I'm going to go through this pretty quickly. They've got way too much here. Again, here's another one of those pathways that you are not going to be required to know, but when you smell something, smell can be tied greatly to emotion, like you smell cologne or perfume or something, that'll trigger an emotion. And ultimately, this will be interpreted in your temporal lobe, what that smell actually is. There you see cranial nerve 1, the olfactory bulb, olfactory tract. I'm going to skip through a couple of these. The sense of taste, we covered that actually, I think, last semester, but I'm going to give you a little taste map here. In fact, they may have it in here. These are chemoreceptors again, bipolar neurons. They don't give you a map here. So let me do this on the board real quick. Usually here's what you see. Some version of this. Some of these may even be kind of superimposed on each other, but so this is your taste map. Salt and sweet more on the tip of the tongue, sour on the sides, and bitter in the back. And bitter in the back, you know, all I can say is the great engineer knew what he was doing, because a lot of your poisons, for example, are bitter tasting. And if they're tasted on the back of the tongue, you also have the gag reflex, which is aided by not only the vagus nerve, but cranial nerve 9, and that will cause you possibly to gag those back up. And then there's one other taste. If we were doing this in person, I usually give a bonus article on this to look up, but with you guys, I usually give a completion bonus at the end of the semester if you make it through the class. But umami or umami is supposed to be the perfect combination of the 4 other tastes. So if you ever watch the Food Network, they're always talking about that term, umami, because they're wanting to make a dish that you can taste the sweet, salt, sour, and bitter all in the same dish. Personally, I think that would make me want to puke anyway, but that's what they're trying to accomplish. This is also something that this basically brought on the discovery of MSG by an Asian biochemist. He would eat something his wife would make, and he loved the taste of it. It was addicting to him, so he actually extracted the amino acid salt glutamic acid and made MSG out of it. And it's in so much of our food. You should really look at labels, and you should look that up and see what you're eating, because it is addictive. A lot of experts think that that's why we have such an advent of diabetes in this country, solely because of MSG in our food. Oh, one other thing, if you eat something spicy, it can actually cause pain. This is something that's always been a little bit of something I can't grasp, because I can't handle real spicy foods, but I've seen other people just eat it and it doesn't bother. So this is probably selected for the individual. All right, let's skip on up. There is a question I sometimes ask on the test, and I didn't used to give a lot of hints about this. I would basically have students rely on their review skills. But if you remember from 137, when we did the cranial nerve study, the facial nerve carries the sensation of taste on the anterior two-thirds of the tongue. The glossopharyngeal cranial nerve 9 carries the taste sensation on the posterior one-third of the tongue. So think of the taste sensations you have on the anterior two-thirds, and then the one taste sensation that is back on the posterior one-third of the tongue. That's a big hint for something I might ask on the test. All right, moving through sense of hearing. I would like you to know these 3 functions of these 3 structures. The auricle, which is basically like a funnel. The auricle is this part of the ear, not necessarily this. This is called the pinna. You see that mislabeled in a lot of text. And that's going to funnel it into the external acoustic meatus, or auditory meatus, which is going to carry that into the tympanic membrane, which will vibrate in response to that. So know those structures, the functions. These 3 do not know the functions. And then I want you to know the function of the eustachian or auditory tube and what it does, and it is right here in this verbiage. All right, let's go on here. I'm going to move rapidly. These reviews take a long time to upload, so I'm trying to keep them as brief as I can and as direct as I can. Okay, as far as the hearing mechanism, I would like you to know the cochlea is what functions in actual hearing. There's actually something in there called the organ of Corti. I will point out that slide when I get to it. That is the neurological structure of hearing. And the semicircular canals and vestibule have to do with balance and equilibrium. If you remember, that's tied to cranial nerve 8, the vestibulocochlear nerve. There's the name, the organ of Corti is the actual specific organ of hearing neurologically. Again, these are bipolar neurons. I'm going to move forward here. We're going to get to the sense of vision last, I believe. So any of this that I'm skipping through, a lot of these you probably don't even have in your PowerPoints because I've reduced them down. All right, the sense of vision. Primarily what I want you to know on the sense of vision, I'm going to say this in this review because I usually do it in the in-person class. This particular diagram will probably show up on your first lab exam. This is a lecture review, but that could be on the lab exam just simply to label. You notice the lateral rectus is cut. I have a diagram that may be on there where this is not cut, but the easy way to learn these, rectus means straight, or has some relationship to a muscle being straight, like the rectus femoris that we covered in 137 that goes straight down the quadriceps, or in the quadriceps group. So you have 4 straight muscles: superior rectus, medial rectus, lateral rectus, inferior rectus. And then you have 2 that turn on a tangent and attach to the eyeball, and they're called obliques: the superior oblique and the inferior oblique. You can see they kind of turn. And they actually move the eye opposite of what you think. The inferior oblique will actually turn the eye upward, superior downward. You don't have to know the actions, and you don't have to know which cranial nerve supplies these, but remember you had cranial nerve 3, 4, and 6 all move the eyeball. Cranial nerve 3 will actually innervate 4 of these, cranial nerve 6, 1 of these, and cranial nerve 4, 1 of these. But all I want you to know is to identify them on the lab. So I always take that opportunity when I'm reviewing to say that. Okay, as far as the eyeball, and this will be one of the diagrams for the lab exam, but in the lecture exam, I want you to know what composes each one of these tunics. Tunics are layers. This is a biblical term as well. The Bible says if someone asks for your cloak, give them your tunic too. So that means a layer of something. So I'm going to use these terms: fibrous, vascular, nervous. As we go forward on the slide, it will say these 3 terms, but I will use these. And I'll remind you as we get to the slide. Okay, so the outer, I will say fibrous tunic on the test. I just want you to know what's in it: the cornea and the sclera. These are both dense connective tissue. The cornea is the front part of your eyeball that feels like you have a 2x4 on your eye when you get a little speck on it. And then the sclera is in the back here. Again, that's more lab. We'll go over that in the lab review. But just know that those are in the fibrous tunic. The middle tunic is the vascular tunic. I want you to know that the iris, ciliary body, and choroid coat is in there. The iris is the colored portion of your eye. The ciliary body, right here, that holds the suspensory ligaments that attach to the lens. And then the choroid coat is in the back. If you've ever dissected a cow eye or sheep eye in high school, and you peel that retina back, you'll see the choroid coat looks really almost metallic. That's because there's a high degree of blood in there, and that's probably how the formaldehyde reacts with that. Gives it kind of like an aluminum foil type view to it. Okay, don't always put this on the test, but aqueous humor is in the anterior chamber of the eye. That is blood plasma. We've called blood plasma several things already. In the skeletal system last semester, it was synovial fluid, that's blood plasma. Cerebrospinal fluid, that's blood plasma. Aqueous humor, that's blood plasma. So, lymph, when we get to the lymphatic system in this semester, or in this class, lymph and tissue fluid are both basically blood plasma. So they should call them that, probably. The lens is biconvex. We'll take care of that more in the lab. It is just concentric rings of protein. So protein can be denatured, as we know from biology 137. So what happens to the lens, it's like frying an egg over many years. It gets cloudy. So when they do the surgery, they pull out that protein and they put what they call a fixed diopter glass lens in there. It's 20 diopters. So if your grandma or someone has cataract surgery on both eyes, they really shouldn't need glasses to read. They should have 20/20 vision, except maybe close up they'll still use reading glasses. But that will give them 20/20 vision. Okay, I'm going to skip through several here. I would like you to know from last semester, it's kind of a review thing, and I love the nervous system, but the sympathetic and parasympathetic effect on the iris of the eye, which we're talking about your pupil sizes. Your pupil big, is your pupil small. What I tell my in-person classes, sympathetic nervous system is the fight or flight. You're running from a lion. So if you're running from a lion, do you need to see more or do you need to see less of what's in front of you? You need to see more, so it will dilate your pupil. Parasympathetic nervous system, more homeostasis, it will constrict the pupil. So that's something good to know for the exam. Aqueous humor is a gel-like substance found in the back posterior chamber. I'm sorry, that's wrong. Aqueous humor is we just talked about. I'll mention the vitreous humor as one of those. Okay, finally, after all that, we're in the third tuning. Don't forget, we did a fibrous, we did a vascular, now this is the nervous tuning. And the retina is what is in there. The retina is a direct extension of cranial nerve 2, the optic nerve, into the back of the eye. A couple other things that are good to note on this slide. The fovea centralis is the center of the vision. If you have a light beam coming into the eyeball, you want it to converge right here in the fovea centralis. If it converges behind that, you're farsighted. If it converges in front of it, you're nearsighted. We're not going to go into that on the exam, but there's also, we'll talk about this in slides later on, but there's more photoreceptors, which we mentioned in one of the first slides of this chapter, for color, which are called cones. They are basically centralized right there in this fovea centralis. So you have the most concentration of these right in the center of your vision. The optic disc is a blind spot. I should have shown you that on the last image, but I'll show you both of these on the last image here. Okay, the optic disc is a region here. It's not necessarily a structure, but it's where the optic nerve, the blood vessels, the nerves, they're coming into the eye, so there's no visual acuity, there's no sense of vision there. So it's a blind spot, and that is why everything is entering there. So on the lab test, we'll probably just point to that area and ask what that area is called. The vitreous humor, while I'm here, is a gel-like substance in the posterior chamber of the eye, and its sole purpose is to keep the retina flattened against the posterior eye, so it doesn't wrinkle, become detached. All right, let's go on here. A couple more points that we're going to get out of this chapter. I want to try to keep this brief. We have the heart to go on this, and that's going to be the biggest. So let's go all the way up to rods and cones. Rods have the sense, they say colorless vision, it's black and white. These are concentrated on the periphery of the retina. So if you look peripherally when you're in the dark, you will see better because there's more concentration toward the side of your eye. Cones, as I said earlier, concentrated in the fovea centralis. These are for color vision. And I think that's it for this chapter. Those are some key points to know for the exam. So let's go on and look at chapter 15. So what I'm going to do in chapter 15 is I'm going to make a separate video that you can access on YouTube about the blood flow inside the heart, what we call the they usually call it the pulmonary blood flow, but it's also the systemic blood flow. But we're going to trace a drop of blood from the right atrium back to the right atrium. And I'm going to do that on the board separately, so if I refer to the blood flow, that'll be on another video that I post on this review. But when we start this chapter, we start by defining the pulmonary circuit is the right side of your heart, systemic circuit is the left side of your heart. Pulmonary refers to lungs, that's blood being pushed to your lungs to be oxygenated, to put oxygen in it. Systemic is going everywhere else. And not to confuse you, but this also goes to the lung, but it is to supply the lung with blood. So this sends blood to the lung to be oxygenated, this sends blood to the lung to keep it alive. But right side, left side, get that in your mind, and when I do the flow, we'll go through that systematically. So a lot of this, I'm just going to say, refer to the flow. If you learn the flow of blood through the heart that I'm going to diagram in that other video, it will tell you a lot because I'm going to test on some of that on the test about the flow. Again, you have two upper chambers, two lower chambers. I'm just going to put that video on separately. That's the first thing I do when we go over chapter 15. We jump in with both feet, and it's really going to help you answer a lot of these questions. So use that video in concert with this review to study for your first lecture exam. The heart lies between the second and sixth rib. Now, let me say that again, second and sixth rib. I have students say, yeah, but the book says, it says the second rib and the fifth intercostal space, which is between the fifth and sixth ribs. So between the second and sixth rib, it's slightly left of the midline. This is why you have 2 lobes in your left lung and 3 in your right, because the heart actually makes a cardiac compression, takes up some of that space. It's about the size of your fist. What I tell students is, look at the size of this fist, and if you accuse me of not having a big heart, then you're wrong, because my heart's about the size of this thing. So you may think that when you're studying for tests, but it's not true. It lies just above the diaphragm. Sometimes I think it actually lies on the diaphragm. The diaphragm is obviously separating the abdominal cavity, abdominopelvic from the thoracic, but the heart almost lies on top of it. Sometimes you can get pain in your heart from gas in your stomach. Feels like you're having a slight heart attack. There's a good picture of the position of the heart. All right, let's go on. A lot of these you don't need. Okay, you need to know these 3 coverings. And we actually covered one of these in 137, so it should be a review, but there's actually 3 names for one of these. But we're going to start, we also talked about the parietal and visceral pericardium in 137, and we talked about visceral parietal pleura, we talked about visceral parietal peritoneum. The lung, for example, is directly covered by visceral pleura. Visceral means pertaining to the organ, so it covers the organ directly. Then we said parietal pleura actually lines that pleural cavity. In the case of the heart, it's a little different. It doesn't line the mediastinum where the heart sits. Again, the heart sits in the mediastinum. That was from 137, which is behind the sternum, in front of the vertebra, and between the lungs, that space. But the parietal pericardium does not line that space. The heart is different. In fact, I'm going to draw this out here. We'll go over here again. Just real quick, I think this is about as easy to show these. So we get different colors here. And some of these may not show up the greatest, but we'll try. So if here's your heart, the visceral pericardium is going to cover the heart directly. Now, if there's a space in here, it's not intentional. I'm just not that good of an artist. But that is your visceral. Again, pericardium, which covers the heart directly. Well, then out in here, hopefully you can see that red stripe. It's not real visible. We're going to call this the fibrous pericardium. Now, what the parietal pericardium does, I grabbed the blue pen here. The parietal pericardium actually lines the inside of the fibrous. And again, this should be adhered right to it, but I'm doing this for effect also. So what we end up forming here is the combination of these form the pericardial sac. So there's a sac around the heart. This one being the parietal pericardium. So in here we have serous fluid that lines this. And if you get too much fluid in there, it's similar to the brain. The brain only has so much space, so if you get a tumor in there growing, it might be okay for a while, but then you get out to the bone and it starts compressing the brain. Same thing here, you get too much fluid, there's a little tiny bit of stretch here. But the fibrous is very fibrous connective tissue, so it's not going to stretch a lot. So if you get too much fluid, then it's going to start putting pressure in on the heart. We call that condition cardiac tamponade, which happens when you get too much fluid around the heart. All right, that's probably not the best picture now, but hopefully that gives you the idea. But you'll be responsible for those on the test. Now, next we're going to talk about the layers of the heart in the wall of the heart. So if we'll start from outside to inside. Outside we have epicardium. Epicardium is the same, also known as visceral pericardium that I just drew here. It is also known as the visceral layer of serous pericardium. So we have 3 names for this. Epicardium, visceral pericardium, or visceral layer of serous pericardium. The middle layer is the muscle. Anytime you see the prefix myo, that pertains to muscle. Myocardium. Myocardium is the thickest layer of the heart by far, and we'll talk a little bit about that. It's thicker on the left side than the right side. Finally, endocardium is like an endothelial layer on the inside, very smooth inside the heart. Everything should be smooth in the vascular system, hopefully, unless it gets unhealthy. All right. This is going to start the flow, and I'm going to cover that in that other video. So again, use that other video, because I'm going to talk not only about the flow, I'm going to talk about fetal circulation, blood pressure, and some failures of the heart in that video. It'll give you a lot of information that'll be on the test. Okay, a lot of these pictures we're not going to use, so bear with me here. These are all, we don't do dissections in the in-person class anymore because it all looks like the same brown color. Let's see, I do want to make a point here. I'll probably do it on the other video, but we talked about myocardium. This is myocardium in here, the muscle layer. Notice how thick it is on the left side versus the right side. I'll cover this in the video, but the left side has to overcome more back pressure when it pumps out. These pump simultaneously, they contract simultaneously. The atria up here and the ventricles here contract together. You know, these two contract together, these two contract together. So when the bottom chambers are contracting, this one is having to push out with more pressure coming back than what the pulmonary side. Remember, right side pulmonary, left side systemic. I'll cover that in the other video. Okay, well, let's get through these pictures. Skip through these pictures. We'll cover the flow through the heart. This is a good thing to know, I guess. This is a written flow through the heart. I think it's a little incomplete. Again, I'm going to cover this in the other video about blood flow. But blood from, might underline that, the systemic circuit, so all over the body, comes back to the heart through the superior vena cava, inferior vena cava, and coronary sinus. When they spell vena cava like that, it's plural. But it's superior vena cava, inferior vena cava. Into the right atrium, through the tricuspid valve, which is also known as the right atrioventricular valve, into the right ventricle. It goes up through the pulmonary semilunar valve, which pulmonary valve is becoming very common to say, semilunar valve, into the pulmonary trunk, which splits into the right and left pulmonary arteries, and goes to the right and left lungs, respectively. From the right and left lungs, we now have oxygenated blood coming back via the pulmonary veins into the left atrium, excuse me, right and left pulmonary veins, and that will flow through the mitral valve or left atrioventricular valve, or also known as the bicuspid valve, into the left ventricle, through the aortic semilunar valve, into the aorta, and then to the systemic circuit where it's going to give up that oxygen. So that's a good written flow. It's a little incomplete, but I'll cover that in the other video as well, that flow. And I won't ask questions about the flow. Once you know the flow of the heart, blood through the heart, you know a lot of the anatomy, and it's going to help you tremendously. Okay, this is a pretty busy slide. Again, the primary thing I want you to know here is that the heart is a selfish organ. It immediately will throw off the first two branches it throws off after going up through the aorta into the systemic circuit is supplying itself, the right and left coronary arteries. If one of these gets clogged up, you can have a problem, especially this one, because it's going to immediately branch into these two. This takes a little longer to branch. The left coronary, a certain area, is called the widowmaker if it's clogged. But I want you to know these are the first two branches, and that it ultimately is going to return via the coronary sinus. That is draining blood from the heart itself. And again, watch the other video. It's going to be very informative. All right, so let's keep going here. The other video is going to have a lot of information in it that will be pertinent to the exam. Okay, heart actions. The term systole means contraction. The term diastole means relaxation. So when you have, blood is going to fill the top chambers simultaneously, and as it's filling the top chambers, they are in diastole, they're relaxed. Once those are full, we open these valves, which I'll cover in the other video, and blood will drop basically, most of it via gravity, but then there's a 25 to 30% that those atria squeeze, and they do that together. Even though the right side of the heart pumps blood to the lungs, the left to the system, we also have the top chambers working together and the bottom chambers working together, which is very interesting. but they don't conflict. When these two when these are in relaxation, these have contracted. So if these are relaxing, these are contracting. If these are relaxing, those are contracting. This is where we get your blood pressure. It's probably a good thing to know too. 120/80. This is millimeters of mercury is our measurement. The top number is called the systolic pressure. The bottom is the diastolic. This is where we get these names. So this is the maximum pressure that the heart has to contract to overcome the back pressure on the system when it's contracting. This is the maximum pressure when it's relaxing. We'll cover that later in the chapter, I believe. All right, the cardiac cycle, I don't know that this is in the notes anywhere, but the cardiac cycle lasts between 0.8 to 1 second. What does this mean? That means if my cardiac cycle is 1 second long, my heartbeat is 60 beats per minute. Average heart rate's between 60 and 80 beats per minute for most people. If it's 0.8, that's closer to 75 beats per minute. So somewhere, if you have a quicker cardiac cycle, that means your heart's beating faster. If it's slower, then you have lower beats per minute. I'm going to let you actually read through this. I think this is a good accompaniment to the other video I'll post on the flow, and I'll let you read through that to prepare for the exam. It kind of takes you through step by step. Let's go on here. Let's go on. Okay. We're going to try to build up a little bit to the 8 EKG, and again, my review is never this long, but I guess I'm lecturing a little more than where I need to. But you need to know that the word syncytium, that the heart works as a syncytium, and just take the first 4 letters of that word, sync. It works in sync. We don't want the top right and the top left beating at a different time or contracting, and we don't want the bottom doing that either. So it has to be in sync. And how we do that, the heart has its own conduction system. It's called the cardiac conduction system. We have little groups of cells that have what we call autorhythmicity, meaning they can beat on their own. But that does not mean they can beat without the brain telling them to beat. But they do beat on their own with the help of the nervous system. Some of this will be lab material. I'll just take you through this real quickly. One of the collections of cells is called the SA node or sinoatrial node. We have an atrioventricular node or AV node. We have an AV bundle. We have a right and left bundle branch. And then these fan out into the myocardium as Purkinje fibers. So what happens here, and there's a good video in your PowerPoints, but I can never play it. They've changed some software. But I disagree. It's a great video, but I disagree with one of the things they say. The SA node is called the natural pacemaker. It will cause the heart to beat about 60 to 80 beats a minute. It can wear out. If it wears out, the AV node will take over, but it will cause your heart to beat at 50 to 70 beats a minute. And then the AV bundle, which is also known as the bundle of His, will cause it to beat 40 to 60 beats a minute. So this is kind of as your heart wears out, something else takes over. But this is the natural pacemaker. It sets the pace, 60 to 80 beats a minute. So what happens, you have an action potential or nerve impulse from the brain that's telling this, and it's going to send it out to the right and left atrium. The problem is, the right atrium's right there beside it. The left atrium's over here. And here's where I disagree with the video. We want these to contract at the same time. So if you remember depolarization in chapter 10 and 11 last semester, we have a series of voltage changes. The heart depolarizes, then it contracts. So we want these to depolarize and then contract simultaneously. So as this is sending this over here, there has to be a delay. And you'll notice some fibers called a junctional fiber. Those actually slow down the nerve impulse getting to the AV node. So this can catch up, and then those contract simultaneously. Once it gets to the AV node, it speeds up through the AV bundle branches, and out into the Purkinje fibers. It goes. But in this case, it slows it down so the left atrium can contract in sync with the right atrium. That is the cardiac conduction system. Okay, let's go on here. The EKG is just a measurement of these basically electrical activity. It doesn't necessarily tell you if someone's alive or dead, but a good cardiologist can look at that in 5 seconds and say, oh, you've had a heart attack because you have an increased ST interval. They know all that, they've learned that. But I want you to know what each one of these signifies. The P wave is when the atria are depolarizing, then they will contract. The QRS complex is when the ventricles are depolarizing, and then they will contract. The T wave is when the ventricles repolarize. However, there's one thing missing here, and they never put it in the text. We also have atrial repolarization. They have to get ready to contract again. They have to go back to that resting potential. So it is actually hidden. Atrial repolarization is hidden in this QRS complex because it's such a dramatic change here. And I'll show you this. This is not a normal EKG wave, but I think they've done this for effect. But here's your P wave, this little blip here. If you can see my mouse pointer. Maybe you can't. This is the P wave, atrial depolarization, then you'll have contraction. This is the QRS, ventricular depolarization, a lot bigger event. Then you have the T wave, which is this little blip, which is atrial repolarization, ventricular repolarization. But lost in here is atrial repolarization because this is such a large event. This is a more normal looking wave right here. In the movies, when someone flatlines and they shock them back to life, that's really doesn't happen a lot. They're probably dead. But talk to your cardiologist, he would know more than I would about that. Okay, skipping these. Okay, what regulates the cardiac cycle, this 0.8 to 1 second? Exercise. If you're exercising, what do you think your heart's doing? It's beating faster. So your cardiac cycle is actually decreasing, making it beat faster. Body temperature in general will also speed the heart rate, especially if you're big like me. If the temperature gets over 70 degrees, I'm going to probably get a little increase in heart rate. The ions, this should make sense based on depolarization, repolarization of the heart, or of anything that we learned last, of the nerve, that we learned in Biology 137. Sodium could be put in there. If you're decreased in those, you cannot either depolarize or repolarize the nerve nor the muscle. You should know these for sure. Parasympathetic, again, is going to decrease heart action. It's about homeostasis, slows the heart. Sympathetic speeds the heart. And this is all controlled in the medulla. That's a review from last semester. The medulla oblongata, or just simply medulla, with the cardiac center in the medulla regulates this. Okay, let's see. One way we regulate this, we have baroreceptors, which are constantly monitoring your blood pressure. There are some in the carotid artery bifurcation, which we call the carotid sinus, and some in the aorta. So just listen to this. Hopefully this will sink in. If my blood pressure is low, what is the outcome that we want to make it increase? Based on the autonomic nervous system, parasympathetic or sympathetic. So if my blood pressure is low, I want something that's going to make it higher, which is going to be a sympathetic response. So these two areas are going to feed back information to the medulla, and ultimately you will have a sympathetic stimulation back to increase the pressure, the heart rate, the stroke volume possibly. The other scenario, your blood pressure is too high. What is this going to do? It's going to send that signal again to the medulla. The medulla is going to send something, probably via the vagus nerve, parasympathetic, to calm it down. So I may ask you a question about that. What happens if your heart rate, blood pressure is too high? We want to calm it down. Or if it's too low, we want to speed it up. Okay, I'm going to get through this. Last section here, blood vessels. We're going to consider the artery, capillary, and vein. Because arterioles are small arteries and venules are small veins. So we're going to just talk about more of the anatomy here. Arteries and veins, to compare and contrast, they all have 3 layers, except the capillary, but the artery and vein has 3 layers each. But you can see on this is the artery, this is the vein. There's a couple things that are different here. Specifically, the three layers are the tunica externa, or adventitia, the tunica media, and the tunica intima, or interna. The tunica externa has a lot of collagen in it, connective tissue. Tunica media is the muscle, the smooth muscle. And the tunica interna, or intima, is an endothelial line. So if you notice here, the artery has a much thicker muscle, tunica media. That is because arteries are carrying blood under higher pressure. The vein also has a specialization in its tunica interna, or intima. It has little flaps that act as valves. And those are very important in returning blood back to the heart. And we'll talk about that. You don't need any of these pictures. Again, you can look in the notes.